So the purpose of this lecture is, so we said many times during the week that assay libraries are important in targeted proteomics. This has been a theme. And on Monday, Ariel mentioned a bit in sort of brief terms how you might do this for SRM assays. And I think on, well, one of the days, tutorial 3, you actually built some spectral libraries in the tutorial, but we haven't really gone into very deeply into the detail of where these come from, and this is really the purpose of this talk. So this is just to remind me to say that this procedure that I'm going to kind of talk about is written from the perspective of making libraries for DIA or SWATH, but conceptually and actually procedurally, it's almost exactly the same as in SRM or PRM. And so I will say SWATH a lot during this talk, but I think that you can kind of equally conceptualize this for the other methods. It just happens to be the case that when we started doing SWATH analysis, things went kind of larger scale. We started paying a bit more attention to this, and we started kind of looking into the details and doing a bit of optimization and so on. And so this is kind of the perspective that I come from. But I think it's important to realize that it's very similar to what you would do here. So, okay, here's the outline. So why do we care about assay libraries? Again, I think this might be slightly repetitive, but we'll just go through it quickly. And then the largest part of the talk will be kind of a walkthrough, a procedure that we developed for doing assay library generation, and I'll try to point out a few key issues. And then I'll mention some of these publicly available resources that we already kind of talked about during some discussion in previous talks, and maybe a few comments about library portability, assay library portability across different experiments or instruments and so on. So why do we care about assay libraries? So here's this is a SWATH-MS2 spectrum, same one I showed yesterday, collected from 600 to 625. This is the precursor isolation window, and so you remember that what we see is a kind of quite complex mixture spectrum where we can't use classical DDA analysis, for example, to analyze this data. And I've highlighted again the major fragmentines for a particular peptide that we happen to know that's in there, and just to point out that these are kind of not the major ions of the spectrum, right? So a classical DDA search engine analysis doesn't work, and so what I said is we need some way to somehow cut through the complexity of this spectrum, and the way that we do this is by building in some prior knowledge into the experiment, right? So we say if we already know something about this peptide and we know what the fragmentines are, maybe we can kind of dig these out of this signal. So here's the library spectrum for this peptide, and once we now know the library spectrum, we can do extracted ion chromatograms of only the parts of the spectrum that we're interested in, right? So extracted ion chromatograms of this ion, this one, and we get, if we plot this as a function of retention time, these intensities, we get back to this kind of pseudo SRM data, or what I talked about yesterday in the SWATH talk. So this is and our goal here is now to get this part of it, right? So we said how we do this yesterday, and Yancheng will talk later on about how we do this type of analysis at large scale, but my concern is where this library information is coming from. And so this picture you've also seen several times this week, so it's the same assay library basically as what you had for SRM analysis, but now we sort of kick out some parameters that we don't need. So the instrument-specific parameters like the dwell time or the collision energy, these are not required for SWATH because we're doing a comprehensive and unbiased method. So we get left basically with everything else. Some information about the fragmentines, their intensities, and the peptide retention time in this normalized IRT space. So this is the goal. This is what we want to get to by the end of the procedure, right? So, and a question we heard when we started doing this type of swath experiments a few years ago was, well, so Rudy would stand up, or we'd go to a meeting and we'd say, okay, if you collect some swath data and then you have an assay library, you have some prior knowledge about the peptides that you're interested in. If you kind of put these two things together in a targeted analysis, then you get nice quantitative data. And everyone would say, well, where do these assay libraries come from? I think there was a lot of confusion about this. And I think that people who had done some SRM, for example, kind of understood this a little bit, but maybe the more DDA-focused people weren't quite sure about what this meant. So what we decided to do was to write this down, actually. We decided to basically describe this procedure of how to build high-quality assay libraries for targeted analysis. And we just wrote it down in a procedure type of way so that people could kind of follow this. And I mean, the goals here were two things. So we had some ideas about what we thought was what the best practice should be for people to use, and of course there are many ways to do this, but this is just our suggestion of what we think is the best way. And we also, because we started to distribute now some of our libraries publicly for other people to reuse, we wanted to kind of document how these were made. So let's get into now the actual procedure that we're using, and I'm just going to try and walk down through this procedure and say what things are at each step. This is, of course, it's basically, I mean, really the same. It's very similar to this protocol paper, but there's much more detail in the paper, obviously, and I'm just going to try and point out a few things that we think are important. So in a simplified way, what the procedure looks like is we have some kind of DDA data acquisition in classical shotgun experiment. And when I asked at the start of the week who's familiar with this, I think almost everybody was. So I think this part you should understand. Data conversion and peak picking. This is kind of an often ignored part of the procedure, but going from the profile mode spectra that we collect to a centroid mode spectra, this can have some effect on how the library looks like, and I'll mention what this is. Then database search, this I think you're probably all familiar. This is Mascot or Sequest or something, or one of 20 other search engines. We think that controlling the quality after this database search is pretty important, so I'll say something about this sort of false discovery rate control of the search results. And then what we need to do is combine all these search results into a spectral library so that we have only one representation one spectral representation of every peptide that we're interested in, and then we essentially filter the spectral library to get to the assay library. So we decide which transitions we're going to monitor and so on. And so this is basically what we want to do, and this is the simplified version. If you look in the paper, you get a diagram which looks a bit more like this, and this now looks kind of pretty complex. But if you break it down into little steps, then it's not too bad, right? So you have some kind of sample prep and data acquisition. We have the raw data files that come from the instruments. We need to choose whether we're going to convert this into different file formats, or more importantly, do peak picking actually. And then you need some kind of search engine. We're typically using these open source ones. We also use these tools from the TPP. So TPP is Trans-Proteomic Pipeline. If you're not familiar with this, this is a set of tools developed at the Institute for Systems Biology in Seattle, which are focused, well, to a large extent on peptide profit, protein profit, and iProfit are the sort of core tools in this library, and these relate to kind of estimating errors in DDA search data and kind of doing protein inference and combining search engines and so on. So we rely here a lot on these TPP tools. There are of course other tools available, and you can sort of use whatever search engine you want, but this is the one we use. We also are doing this FDR control of the search engine, the DDA search results, using a tool called Mayu, which is also distributed from the TPP. And the tool that we use for spectral library building is called Spectras. This is also in the TPP. And then finally, what we want to get to is this table, right? This assays in table format, which will then be usable by any one of these softwares. Like Skyline will import this table, but you'll also hear about Spectronaut from Lucas tomorrow, or PeakView, or the tool that was developed in our group here, OpenSwath, which Yansheng will talk a bit about in the afternoon. And so and the idea is to get either this kind of CSV based table, or there's another file format called TRAML, which is an XML based format from the Protein Standards Initiative, which is kind of sort of a specific standard for exchange of these like assay libraries, which is used by OpenSWATH. And then you can either add decoys at this stage if you're using OpenSWATH, or some of these tools like Skyline, as you saw, will add the decoys internally themselves. And so, yeah, I want to point out that this might look a bit overcomplicated or very detailed, and it is kind of, it's certainly yeah, it's many different tools, and you would have to become kind of familiar with all of these, but it's all written down in the protocol how to do this. But I just want to point out that there is certainly a much easier and faster way to do this, and you can use your favorite search engine, whatever you're using now for this part, right? And Skyline will do, so Skyline, as I think Ariel mentioned, has a spectral library tool coupled with it, which is called BiblioSpec, and this will do more or less the same thing as what SpectRust is doing here, and the filters in this peptide settings and transition settings will do a similar thing for filtering the set of transitions that you want. So there's certainly alternatives here, but what I'm going to discuss is this on the left side. So at least from a conceptual perspective, you understand what's happening at each stage, and then you can kind of make your own decision about what you want to use here. So yeah, I mean, Skyline, you can either use the assays that you generate with this workflow in Skyline, import these into Skyline, or you can use Skyline to do this kind of second part of the procedure. And Brendan already mentioned that this is compatible with like 20 different search engines. He showed a list, I think, and so it's likely that whatever search engine you're using is probably supported by Skyline. Okay, so let's go down through this. So, I mean, the sample preparation and data acquisition, the question is what kind of samples do we want to use to do this spectral library generation? Well, I mean, the main point is that the targets that you're interested in should somehow be in this sample and should be accessible in a DDA analysis. So this can be samples from kind of different experimental conditions. So if you have a perturbation, maybe you want to measure samples for the library that come from, you know, both or all parts of the perturbation, such that if a protein is only expressed in one condition, then you have a chance of having it represented in the spectral library. We also have done a lot of fractionation to extend the depth of the spectral library. So many people would do like off-gel separation or strong cation exchange, or I think Lucas maybe will talk about some high pH reverse phase fractionation, for example, to build larger libraries. Or the other alternative is what we've already heard about, is to synthesize synthetic peptides or recombinant proteins. I think recombinant proteins is a great way to do it but it's hard to do at kind of large scale. So there's a paper from the McCost lab saying you can use like in vitro transcription translation systems to express the protein, and then you just digest it and put it in the mass spectrometer, and that way you get information basically about all the peptides that are detectable from the protein, whereas you might not get that from synthetic peptides. And in this type of when the samples are kind of so-called real samples, real biological samples, what you'll get is hopefully the best peptides, but that's not necessarily always the case. So this recombinant protein idea, I think, is quite nice. Add IRT peptides, some kind of standard peptides to normalize the retention time space is valuable. I think Ariel talked a lot about this in depth. But we basically add this in pretty much every sample we run in the lab that might ever have a chance of going into a spectral library. This is worth doing, or it can be any of the kits. Or Ariel also mentioned that some people have done work on finding peptides that commonly appear as contaminants, for example, like from keratin or from filamentous proteins, or proteins that are highly conserved across species, or trypsin or something like this. And you just need to be able to find them all the time in every sample. And once that's true, and once there's a reasonable number of them and they're distributed across the gradient, then I think it's possible to use kind of endogenous peptides as IRT standards. But we don't actually do that because it's just a convenience to add in these other peptides, and they're always there with a high signal. It's also the case that for the DDA analysis, you may want to change some of the instrument settings in order to get the highest quality spectra. And this is potentially may compromise actually the number of IDs you get, so there's a balance here. So for example, we were working on the SciEx on the triple top 5600, and I think SciEx recommends that you use 50 millisecond scan time in MS2 in order to maximize the number of identifications. But in a QTOF, the signal to noise of the spectrum has some dependency on how long you do the scan for, right? So if you do 100 or 150 milliseconds, you get a higher signal to noise spectrum. And this might be useful in terms of how accurate the estimate of the relative intensities of the fragment ions are. So the higher quality your DDA MS2 spectra are, then this will improve the quality of your spectral library. You may also benefit from having more than one MS2 spectrum per peptide or per precursor. So for example, we set usually the dynamic exclusion in the DDA analysis to half of the peak width, and this allows you to have one spectrum, MS2 spectrum, sampled near the start of the elution and maybe one near the peak where it's most intense, and you probably get a higher signal to noise spectrum. We also think it's useful to try and get the highest possible similarity between fragmentation in the library and the swath acquisition, right? So you know that if you change the collision energy, this can probably change the relative intensities of the fragment ions in the DDA analysis. And I'm not sure if we talked about this, but yeah, I think it came up with Jake. But in swath, similarly in sort of PRM, you or certainly in swath, you can't really do any collision energy optimization because you have this wide precursor isolation window, and there are many different peptides in there, and so you know approximately what the mass is, right? But you don't know what the charge state is. So all we can do is say, okay, we know what the mass is, and what we usually do is set the collision energy for a doubly charged ion, which is kind of in the middle of the window, and this is about the best we can do. But what we can do is actually change the DDA settings to match this collision energy settings, right? Then you get the highest similarity between the DDA and the swath spectra. So this is again something that might compromise a little bit the number of identifications that you get in the DDA analysis, but the resulting match between the DDA library and the swath data will be higher fidelity in a sense. Something that's also kind of turned out to be important has been centroiding or peak picking, and so in some instruments this is done on the fly by the vendor libraries, or you can collect the data in profile mode and use a tool afterwards to pick the peaks, right? And so if you're doing a DDA, if you're doing a search analysis, what you need is a list of peaks, right? So that's what the search engines expect, and you need to do some kind of centroiding to do this, and the choice of which algorithm you use to centroid the data can have an effect. And we've seen this, so I think, so this is a slide which shows basically exactly the same MS spectrum centroided with three different centroiding algorithms. So one is this the SciEx protein pilot mode, this tool that they have. One is MS Convert, which is this open source tool, but it's using the AB SciEx libraries to do the centroiding. And one is this QTOF peak picker, which was developed in our lab specifically to do this task. And you can see that the relative intensities of the fragment ions are actually kind of quite dramatically different in some cases, right? And this comes from exactly the same raw spectrum. So whatever differences here are somehow an artifact of the centroiding. And so if you want to then compare to the swath data, right? So here's some swath data, here's the relative intensities, and we want to compare to the relative intensities of the library. Well, which one are you going to use? And so we found that using this QTOF peak picker, well, in fact, it was kind of developed specifically for this application. So the relative intensities that we have between these between DDA and swath here are in best agreement when we use a particular peak picker. This is kind of an extreme case, but I think it illustrates the point that these peak picking algorithms can sometimes, I mean, for example, this protein pilot one I think is optimized to get maximum maximum identifications. And so they may be doing something additional to just peak picking. There could be some charge state deconvolution happening or something like this, or it could be is it using like the intensity of the peak to do the centroiding, or is it taking the area under the peak to do the centroiding? That's one difference. And this QTOF peak picker is actually taking the area under the profile peak, right? The mass spectrometric peak. It's using the area to plot these, and that's one reason why we think it's in good agreement with SWAT. So these are kind of small details, but if you're interested in going and optimizing to the maximum, then it's something you might think about. So what about the search engines? There's not really that much to say here. I think people basically understand how this works. The only thing I would point out is that something that we do use is multiple search engines, right? So by default, I think we're using like Common and Xtandem, and sometimes we also throw in OMSA and Merimatch to very comprehensive, and these tend to be the open source ones, which you can sort of run on Linux on a cluster, which is what we do. But I mean, it doesn't really matter. It can be any search engine. At this stage, they all perform pretty well. But sometimes combining multiple search engines gives you more sensitivity and potentially more robustness, right? And there are some tools which allow you to do this in a kind of statistically sensible way. So one of these is called iProfit. It's also part of this transproteomic pipeline, TPP. And what you see is what's plotted here is your how many identifications you can get at some fixed at some FDR. Based on decoy counting estimation. And what you see is these dotted lines is all these various search engines individually, but when you combine them all together in an appropriate way, then you get a better performance essentially. So each of these search engines was probably optimized or trained in a different situation, and some are better potentially in certain types of samples or certain species or something like this than others. And so whenever you compare all these different search engines and you rank them, people always get a different answer, right? So it's hard to say which one is the best, but we rather say, okay, well, let's just take a few of them together and then hopefully we get a good answer. What about false discovery rate control? So I think it's interesting to think for a second about if we have an error in the DDA analysis, right, we assign the wrong peptide, how will this affect the SWATH data, right? So let's say we have a shotgun, we have a DDA spectrum, here it is, it's not particularly high quality, and the search engine assigns the wrong peptide sequence. Despite the fact that we made some reasonable threshold, there's still some errors being made, right? And so the question is, we now put this into our assay library and we try to extract data from SWATH. Well, if all of these fragmentines came from a single precursor and that we now try to extract all these fragmentines in the SWATH data, what's going to happen, right? We're going to get a very beautiful looking signal, right? Even though we've now called it as the wrong peptide sequence. So now this error is being propagated into the SWATH analysis, and this is potentially a problem. But there may be another kind of error that can come from the DDA analysis. So for example, you can imagine that there's like a chimeric spectrum, and we know this happens actually really a lot in DDA type analysis. So we have different, even though we have this narrow isolation window of 1 or 2 mass units, we can still co-isolate peptides, and the fragmentine spectra that arises is a chimeric spectra. So we have a blue peptide and a red peptide, and we have a mixture spectrum, and this is now the cause of the DDA search engine. It makes the error because of this chimeric spectrum. Well, what happens when we now go into the SWATH data and try to extract this? Well, actually, in most cases, even though these 2 peptides were co-isolated, in most of the time, they still won't co-elute. I mean, you might get unlucky, but in the majority of cases, the co-elution will not be exact. And in that case, our scoring systems will say, hey, these fragmentines didn't all come from the same precursor in the SWATH analysis. And so in this case, the error will be, it'll get a low score and the error will be resolved, right? But the problem is we don't really know. So in some cases, you can imagine that SWATH is resolving the error from the DDA, but in some cases not. And we don't know what's really the prevalence of these different types of errors. And so the conclusion for me from that is that we better try to minimize as much as possible the errors in the assay lib in the DDA analysis. And so to facilitate this, we're using a tool called Mayu. And Rudy, I think, alluded to this earlier, and Brendan a little bit. What Mayu allows you to do is, based on decoys, it allows you to estimate the false discovery rate, but at different levels, right? So the false discovery rate at peptide spectrum match level, peptide level, or protein level can be very different at a fixed score threshold. And the reason for this, if you weren't aware of it, so in this diagram on the right here, each hexagon represents a protein, right? So there's smaller proteins, there's bigger proteins, and the green ones are essentially the true positives, and the red ones are the false positives, right? So what you see is that the green ones the good PSMs, the good matches, tend to build up on specific proteins, whereas the false positives tends to be kind of distributed across all the proteins. And this has the result that if you use, for example, decoy counting to estimate these false discovery rates, and you use a fixed score, you can have a relatively low PSM false discovery rate and a relatively low peptide level false discovery rate, but the protein level false discovery rate can become inflated very quickly. So we would argue that it's important to compute the false discovery rate at each of these levels and sort of be aware of them. And so this Mayu tool is one way to do that. There are other ways to do that. But so if you look, for example, if we want to achieve a protein FDR of 1%, then we need to use, so on the x-axis here I should say is basically iProfit score, but you can think of this as like a search engine score basically, right? So all the good hits are towards the right, and on the y-axis is the false discovery rate plotted at these different levels. So if we say we want to achieve a 1% protein FDR, then we have to set a very strict search engine score threshold, and the peptide level FDR is actually going to be much lower, 0.2%, and the PSM level FDR 0.08%. And so if you're interested in these kind of issues, there's a paper from Lucas Reiter, now 6 or 7 years old, but this was an important step in DDA proteomics to realize that these errors were occurring at different levels. So this relates to what Rudy said in terms of people reporting a lot of weird stuff in the early years of proteomics, and it wasn't really being evaluated in the proper way. And so our suggestion is to take this into account also at the library stage, right? When you're creating your library, you want to make sure that it's reasonably high quality. Okay, so what about the spectral library? So there are several things that are happening in this spectral library part. So for one, we're doing transformation into this IRT space, right? So we've spiked in these IRT peptides. They also get identified in the DDA analysis, and so we can do this type of linear regression that Ariel showed and project the retention times of our peptides onto this normalized retention time space, right? So I think this is covered already and also in the tutorial. And the other issue is that we need to pool spectra to get a similar. We only need one spectrum for every peptide, right? And usually in a DDA analysis, you get multiple instances of spectra for a single peptide, and we need some way to decide which one's the best or some way to combine these. We also have kind of a peak voting system, so if there are multiple spectra recorded, we would like to kind of take the highest quality representation so we can do some kind of consensus of these. I'll say what this means, but there's also some quality control here. If you have multiple spectra, you can sort of kick out the ones that look weird, right, that don't agree with all the other ones. So what do I mean in more detail by removal of these redundant spectra? So here's sample 1, sample 2, sample 3, and what I plot here is the precursor ion traces, MS1 extracted ion chromatograms of some peptide, right? So here's the peptide elution in sample 1, 2, and 3, and these red dotted lines are where we're getting an MS-MS spectrum, right? So this red dotted line here, this peptide starts to elute, you trigger an MS2 spectrum, this is the spectrum that's collected. And then you have some dynamic exclusion setting, and you know, it ignores this peptide for a while, but then eventually maybe later it collects a second spectrum. And in the next sample, well, it's a bit lower intensity, so it only collects one spectrum. And in the next sample, you know, we have two, so now we have 5 spectra for this one single peptide, and we need to decide what to do with these 5 spectra. So you basically have two choices. and this is one area where the behavior of Skyline is a bit different than the behavior of Spectrast. So what we do is, in the Ebersol lab, is slightly different than what's done in Skyline. Skyline is basically taking the best spectrum out of these 5, right? It says which one's got the best search engine score. I'm going to use that as the best representation of this peptide, and I'm going to put it in the library. However, it's also possible to use all of these 5 spectra, 4 or 5 spectra, and somehow combine these, make an average into these, or what we call a consensus spectrum. And so we keep all the peaks that are the same, the relative intensities are some kind of average over these, and if there's one spectrum in here which really doesn't look like all the others, that might have been a mistake, then there's some quality control procedures to kick this out. It's also possible that these spectra came from, different types of samples, right? So one might have came from a cell line, and one might have came from another sample, like a tissue sample, where you want to combine the library. And so some of these might have different interferences, they're in different backgrounds. So there may be peaks, there may be fragments in there which are in one but not the other. But what you really want to keep is the signal from the peptide. So conceptually, there would seem to be some kind of advantage of taking all these spectra and using them together. And so a colleague of ours in Seattle, Sam Bader, has written a manuscript looking at kind of many of the issues relating to spectral library generation, but in particular he looked at the difference between using the best replicate spectrum, or if you pool 5 or 10 or 15 or 20 or 40, or in some cases you may have up to 100. And he plotted basically some relative score relative to some random extractions based on decoys, I believe. And essentially what he sees is that, the best replicate, so 5 spectra is better than the best replicate, and 10 is slightly better, but then it kind of saturates. So if you have 5 or 10, then it's worth putting these in a consensus, but it's probably not worth having 100. And this is also what I mentioned about earlier on. It may be beneficial to have multiple instances of the same spectrum. Okay, so I think this has been covered pretty well. We spike in these IRT peptides, and then for our target we make a linear regression, and our target peptides can be then projected from the retention time space in which they are collected onto this normalized retention time space. And this really goes to assay portability, right? Moving it from one lab to the other, or from one experiment to the other, or so on. And this basically works, but I think it's been explained to you already, so we can keep going. This is what I mentioned, that you could potentially use endogenous peptides, but I think I also explained this already, but if you're interested in this is the reference to this paper. Sarah Parker was a visiting postdoc in our lab who came with a bunch of data that didn't have IRT peptides in, so she kind of developed this endogenous IRT approach. But there's now a paper published on it. If you want to try it out, you could try out these lists of peptides which are published in this paper. And then the final step basically is to go from the spectral library to the assay library is the nomenclature that we use. So to go from a spectrum to now just a table, right, a list of fragmentines that you're going to monitor. And so you need a set of rules to choose which fragments. And these basically correspond to the rules that are in this like peptide settings and transition settings tabs in Skyline, but we're actually doing this with a script, but it doesn't matter, it's the same thing. So what we sort of recommend is 5 to 6 transitions per precursor, right? This is based on the data that Ludovic showed in the M-profit analysis. We're typically taking 1, 2, or 3 charged ions. We go from 400 to 2000 M over Z, because in general this lower mass range below 350 or 400 has a lot of interferences. We typically take Y and B ions. Well, at least in the case of a label-free analysis, if you have if you have heavy peptides, then this might cause a problem, right? The B ion series won't work very well. Won't work at all if you have peptides which are C-terminally heavy isotope labeled, right? Because the B ions will not contain this heavy isotope, and they'll look the same in the light and heavy in a swath analysis if the precursor is in the same swath window. You can also use neutral loss fragmentations if you want. We typically exclude the fragment ions which are falling in the precursor window. So if we're collecting from like 575 to 600, we typically would not, in the swath analysis, we would typically not use a fragment ion from this range. Does anybody know why that might be? So if we're doing a swath analysis, we have a 25 Dalton window, and this window is like 575 to 600, is there some reason that the transitions at the MS2 level now in the range 575 to 600 might be problematic? Yes? Why?
Precursors that are not fragmented?
Exactly, yeah. So there's many, this window is going to be very noisy because in particular in a QTOF instrument, everything that goes into the collision cell doesn't necessarily get fragmented, and it means there are many unfragmented precursors in this window. And so if you plot it, if you extract a transition from this range and you plot it beside all the other transitions, typically you see it's very noisy, so we usually kick it out. And then for this OpenSwath tool, we convert to this Trammel format, and we also add decoys at this stage, but that's not necessary for Skyline or for Spectronaut or other tools. Okay, so basically this is what we should have at the end. This is what I showed you, and we have either this table format or this Trammel format, and now you're basically ready to import it into Skyline or into OpenSwath and do the analysis. So, I mean, by way of a summary, a few points that we think are important: that the assay library relative fragmentine intensities from DDA should agree with the SWATH data, right, as best you can, or the SRM data or whatever. So watch out for processing by peak pickers. The collision energy is good to match them from DDA to SWATH. We usually run a little bit longer MS2 scans times to get the signal to noise of these spectra up a little bit. Better quality spectra will agree better with the SWATH data, right? And we always use this combination of spectra into consensus spectra if we have multiple instances of spectra for the same peptide. Control the false discovery rate of the DDA data, right, to avoid propagating errors into the SWATH data, and transform the retention times into this normalized space in such that they can be kind of reused between experiments or across one experiment, and that the predictions of the retention time will be more accurate. Okay, so there's like really much more extensive information in this protocol. If you're interested, take a look there. Okay, so what about publicly available resources? This question also came up, and we've started to do a little bit work in this direction. So we realized at one point that we had SWATH project number 1, now we need SWATH analysis number 1, so we need to make assay library number 1, right, based on a DDA analysis in the way I've just described. And then we said, oh, we have project 2 and project 3 as well. So at some point we thought maybe we should sort of combine these libraries together, at least within one species, and not have to redo this every time. And so this is essentially the project that I will describe here. We did this for human, right? So we took basically all of these samples where DDA data had been collected in the lab, and this resulted in about 330 files, right, that we had sitting around in our database. And they came from very diverse sources like cell lines, well, mostly cell lines, but also some clinical samples like kidney, intestine, muscle, lung, blood, blood cells, etc. And also different kinds of preparations. So some, there was like affinity purification happening, some had size exclusion fractionation, some had different fractionations like ion exchange at the peptide level, but we just mushed all this data together into one giant DDA search. We searched it all together and followed exactly the procedure that I just described to you. And essentially this is the results, right? So the pink circle is everything that's in the UniProt SwissProt database, so that's 20,000 proteins. The green is what has protein evidence at the protein level in UniProt, and the blue is what we actually detected. And I think this number is now a bit larger. Oh yeah, this is it down here. So it's 10,000 proteins where we could identify them with unique proteotypic peptides. And this came from about 140,000 peptides, and we essentially took this DDA data and we went through the procedure that I just described and made a really big assay library. And this is kind of referred to in the paper as combined human assay library, but it's referred to locally as pan-human library or something like this. So if I say pan-human library, this is what I mean. And at one point we were interested in checking, right, whether this library that we developed in our lab could another lab use this library to extract information from their SWATH data? So it goes to this question of assay portability. So what we did was, at the time there was not that much other SWATH data published, but we took what was available from the Anne-Claude Gingras lab. She had published some SWATH analysis of affinity purifications, And there was only, because it was affinity purifications, there was only kind of 150 proteins of interest in this study. So we took all of the assays for this 150 proteins, we took them out of the large library, and then we extracted using our library from her SWATH data, right? Collected on 2 different continents on 2 different instruments. And the question was whether we would get the same answer as what she got or not. And so that's what this graph shows here. So the pink and the green is data from Anne-Claude's library, and the blue and the purple is data from our library, right? And the difference here is that there's 2 different conditions. It's like a wild type versus mutant comparison. But what you see is at the level of protein fold change, we don't get identical results, but we get the trend is very similar. And here in these ones where the fold change is very kind of large in the negative direction, this is probably like a presence-absence case where it's present in one sample but not in the other sample. I mean, the other explanation for why we don't get exactly the same results is that we don't have exactly the same peptides in our assay library, right? So she identified for a given protein a different set of peptides than we did. She had higher coverage because she had these affinity purification samples. But essentially, I mean, our conclusion from this was that it basically works. The biological conclusions of the experiment were consistent. And so we now have a number of publicly available assay resources. The Panorama is the one associated with Skyline that Brendan probably mentioned. These 3 are hosted by the Institute for Systems Biology in Seattle, and the CPTAC group, which is this NCI initiative, has its own portal. The one we're using for SWATH libraries at the moment is called SWATH-Atlas, and this is basically, I didn't update this slide, but this is basically the state of it at the moment. So we have this large human protein library. We have now MHC peptide library from a recent paper. We have a very comprehensive library from Mycobacterium tuberculosis, and we have a synthetic peptide library from yeast. And our intention is to kind of continue filling this out with different kinds of libraries as they arise. And so the assay libraries are in this CSV format, this table format basically that I just described, and also in this TRAML format. This XML-based format. You can also have not only the assay library tables, but earlier on the spectral libraries, right? So if you want to choose your own transitions and do the last part of the processing yourself, you can do this. You can also download the raw data and do the whole procedure yourself also if you like. And you can download subsets of libraries, so you can query this for sets of particular proteins and download the assays just for these. So take a look if you're interested. And yeah. So last thing is library portability, right? So if we are using these kind of large scale spectral libraries, can they go across different instruments? Can they go across different labs? Can they are they actually really reusable? So I think some information has been, this has been looked into to some degree for SRM data. So there's these papers out which said, here was an early one which said you can use ion trap data. If you only use the Y ions from ion trap data, then you can use the libraries generated on an ion trap to generate SRM assays. There was also a paper from Albert Hecht's group saying HCD spectra in the Orbitrap look very similar to triple quad spectra, so we can use these for library assays. And the Agilent and Waters, for example, who both made QTOFs and triple quads, always made the argument that if you moved from their QTOF to their triple quad, the spectra would always look very similar because they had the same collision cell and the same ion optics and so on. And I think this is largely true. So there's kind of some work done here to say that you can move these, there's some portability between these different platforms. We looked into this in a little bit more detail. So there was a master's student in our group called Umut Toprak, and he wrote a little tool that allows you to compare spectral libraries. So if you make these spectral libraries in Spectros format, you can input it into this tool, and it's going to give you some kind of similarity score between these two things. So this normalized angle, this is essentially similar to a dot product as you saw in Skyline. It's a measure of spectral similarity. So if you look across these different instrument types, so the Orbitrap Elite HCD, so I'm always comparing to SWATH-MS, right? So DDA to SWATH. So if you do Orbitrap Elite HCD, well, it's pretty good. If you go to Orbitrap Elite CID, so it means like you're doing the MS2 spectra in the ion trap and the collision is happening in the ion trap, not beam type, then the agreement goes down a bit. Same for this LTQ XL, where the fragmentation is happening in the ion trap. And the best agreement we get is from triple TOF shotgun to triple TOF swath, right? This is kind of obvious because it's exactly the same instrument. But so my point is that it's not really a binary choice. You can use this in one case, but not. You can use one instrument and versus the other. There's kind of a continuum of how well these things agree with each other. So this means that essentially you can probably move between instrument types, but you lose some information. But if it's the only information that you have, it still might be useful. But the recommendation in the end would still be to try and collect the libraries on the matched instrument, right? We're getting close to the end now, so don't worry. But what about IRTs? So what about transferring IRTs against different labs? So this is a plot now showing. I mentioned yesterday, and Rudy also mentioned that we did this kind of cross-lab swath evaluation study. And in this study, all the swath data was collected at a whole bunch of different labs, but we used a single library, right, that was generated in our lab. And what I'm plotting here is the distribution of errors in retention time, right? So the delta RT from the predicted retention time to the measured retention time. And in this largish kind of data set of about 20 files, this is our data with our library, right? So it has the lowest error. The standard deviation is 100 seconds. Here is one of the other sites. It has a bit larger distribution of errors, but not by much. And then this is one of the other sites which has a larger error again. But the difference in these errors is not dramatically large, even though we have not identical chromatographic setups. So similar chromatographic setups. So we have 30 centimeter columns, but we're using a packed emitter with a particular phase. These guys are using chip LC with a different phase, although it's a similar phase. So the point here is that it seems like this IRT concept, which we tried to tell you allowed you to transfer assays across labs, works to a degree, right? It works reasonably well. The errors are not that much bigger when you go to a different setup. Okay, so I'll summarize now. So assay libraries are essential for this targeted analysis of swath or DIA data, and Yancheng is going to talk much more about this in detail in the afternoon. We have now written down this procedure in this Nature Protocols paper that you can try out or take a look into if you like. Our recommendation is that you control the FDR in this assay library generation to avoid propagating errors from DDA to DIA. It seems like these assays are pretty portable if you have same or similar instruments set up. So this is this example where we analyzed data from the Zhangras lab and this cross-lab comparison study. Assay portability across the instruments might be possible, but with some care, right? You should evaluate the differences between these instruments. So the fragment time spectra could be a bit different, or the retention times could be different, or similar depending on how similar your chromatographic setup is. Okay, I'm going to stop there. What's the time? It's okay? Are there any questions? Yep. This is not the most kind of exciting topic, I agree, but it's sort of important one. Yeah.
So how much does it take to go through the whole process that you just described? Oh. And how much subject is to person of the operator versus like automatic or something?
Okay. Yeah, so they asked us, this is a bit dependent on how big your DDA data is, right? So in this protocol paper we wrote, we put like a sort of a toy example, which was 3 DDA files. And then to go through the procedure, I mean, you could do it in a day, basically, in a pretty straightforward, not the data collection. Well, the data collection would probably take a day as well for these 3 files, and then another day for the sample processing. If you know how to use all the tools and you're familiar with everything, then it's pretty straightforward. If you have several hundred files, then it's the time that takes to actually measure and search these, right? And in terms of how easy or difficult it is, well, it's not the most easy procedure to follow because there's several different tools in the pipeline. You sort of have to have a little bit of familiarity with each of these tools, and 1 or 2 of them is like one of them. The last one is a Python script, and there might be 1 or 2 command line tools. So it's if you're if you're used to using like graphical user interface software, then it might be a little bit challenging. If you have some kind of local computational support, then it shouldn't be a problem. But again, the alternative is then to go. There is the simpler alternative, right? The whatever search engine, your favorite search engine plus Skyline is also a viable alternative. And it's a very good alternative. There are just some areas where it may not be completely optimal. I think the main one is this consensus versus best replicate library. But the difference in the end between these two is not so huge, right? It's a small optimization.
So why didn't you go for the, let's say, the ABSIX pipeline? So use the protein pilot and then the peak view.
Right. Well, I mean, in our specific case, there are several reasons. One is that we have a sort of one is kind of historical, right? So Rudy's group has been developing these TPP tools for a long time, and we're comfortable with them and think that they work very well, and we know how they work. Whereas Protein Pilot, you don't know how it works, right? It's a commercial tool. This is kind of a concern. Same with the search engines. We tend to use like the open source ones where the math is published and we know what they're doing. Another practical implication is that we tend to have a lot of data, and our data processing, most of it happens on a cluster, and it's easier to run these kind of like the open source ones tend to have like Linux-based. You can run them on Linux, and the GUI like Protein Pilot you couldn't run on a cluster, for example. So there are there are several reasons, but in the end, the question, Spectrast, our spectral library building tool, is compatible with these peptide profit and iProfit tools, the previous step in the tool, and they're compatible with this. So it's kind of a chain, right? And in our hands, this works well. So there's sort of several reasons. I don't know if I answered did I explain myself?
Did you actually compare both? So your pipeline with, let's say, do it one time or twice with the regular workflow, just to see how much better is your workflow as compared to... Because the other one is, at least according to what you're saying, is far much more automatic and more user-friendly, let's call it like this.
Yeah. It's more user-friendly for people, probably for smaller labs, that it's easier for them to use. For a larger lab, it's actually easier, and you're using like pipelines and clusters and things like this. Logistically, it actually becomes easier to do it the other way probably than have every individual person clicking. As to the comparison question, I mean, we certainly didn't do a direct comparison with, for example, Protein Pilot. We sort of took the approach that a lot of optimization had already went into the searching and the search tools and the search post-processing tools, and we kind of tried to do our best job with this essentially. There are some comparisons now out on the actual swath analysis level. So different tools, so not the libraries now, but when you actually go to extract the data using Skyline or PeakView or OpenSwath or Spectronaut, there's now been a comparison paper that says how these compare with each other, and I think Yansheng will maybe mention this later on. But I don't think, is there a comparison of the spectral library generation tools? I don't think so, right? No. But you can try it yourself.
Okay. So in let's say in a 2-day SWOT analysis on human samples, like how many MS2 peaks get assigned today?
From the SWOT, so in our best data, we probably are going, so this is again something Yansheng will cover, right? But I think in our best data with like our standard setup, which is a sort of a 2-hour gradient, we can sort of detect around 50,000 peptides, maybe 40 to 50,000, which corresponds to somewhere in the range of 4,000 to 5,000 proteins. So that's the best we can do at the moment, but what we haven't done really is push the chromatographic side very far. So there are also many people who are doing like 4-hour gradients, 6-hour gradients with long UPLC type columns. And I think if you did this, this would push up the numbers quite a bit. I think Lucas on Friday, he will talk a bit about doing this on with also some kind of longer gradient and using the Q-exactive type instrumentation. And I think they have some impressive numbers from this data, but
what I mean is like you basically get a view on MS2 peak. I mean, or
Yeah.
so how many get assigned? Like, how much room is there for improvement? Like,
I see.
how much is the unknown?
I see.
You
Yeah.
see the unknown, you just don't identify it. So how much is that? Do you have an idea?
That's a very good question. I don't know if we've specifically looked at this. I can sort of try to guess, but I mean, what you could do. So we only see things that we extract in a targeted way, right? But you could, for example, do a feature finding step at either the MS1 or the MS2 level and say how many things are there in there that look like a peptide. And there are papers going anywhere in the range from sort of 100,000 to maybe, with the high resolution stuff, going towards 200,000 and higher. And so if we can assign 50,000, well, I think there's definitely room for improvement. The question is, what are those other things, right? So we filter a lot of stuff out of our library because maybe we don't necessarily need it. So like non-unique peptides or peptides with artifactual modifications, right? So like N-terminal cyclizations or weird modifications that may not be biological and may not be interesting. But there may be stuff that is biological and interesting that we're also missing as well. And I don't think we have necessarily a good feeling for what the proportion of these things are. But I think one research area will definitely be to go in this direction, right? There's now ways to interpret the data, the SWATH data, not in a targeted analysis, but more in a way which is more sort of related to the sort of spectrum-centric sort of discovery approach that you would have in a DDA analysis. So to try and say, well, what's in there exactly, and then later say, can we quantify this? So that's sort of an open question, I think. But we're definitely not explaining all the signal, and maybe, I don't know, you could say half probably, or it would be hard to estimate.
So like in the far future, like it relates to the end of your previous talk where you said like maybe they're both fields are converging, and with the smaller windows, so maybe like in the ultimate pipeline will be something where we combine both and try to find some within discovery approach and then assign a lot other spectra with this.
Absolutely.
Because now, like with today's technology, like what's your take indeed on, I guess it's an obvious question, like on the unknown modifications and stuff like that. I mean, do you see like you will get a spectrum of everything?
So we'll get a spectrum of everything down to the limit of detection of the instrument, right? So this is important to say this. It's not that we get a spectrum for every peptide in the sample. That's definitely not the case. The limit of detection is 4 or 5 orders. The linear dynamic range is 4 or 5 orders of magnitude, and so once it's within that range, we should have some kind of spectrum for this. And there's also issues of ion suppression and so on that you have to take into account, and then there's also issues of selectivity, right? So sometimes maybe we don't recover a signal because it's too contaminated, it has interferences because of these wide precursor selection windows. So I should say, okay, I mean, there's several things to say. As we have been improving the selectivity, right, either by closing the size of the windows a bit. So I mentioned there we went from like 25 down to now we're using like maybe 6 or 8 in the most dense part. This improves the selectivity. That seems to be the case. So we detect an extra maybe 15% of peptides or something. And this can really probably only be explained in terms of that there was an interference in one signal that's now gone by closing this window. Similarly, if you increase the resolving power of the chromatographic separation, then you may also remove interferences, selectivity issues. So I think that's another area where there could be room for improvement. But the question, so in the DDA world, people have tried to, you can ask the same question, right? How many MS2 spectra are explained by our database search versus how many MS2 spectra are there? And it's always a rather low number. It's like 30, 40%, something like this. Maybe it gets up to 50, I don't know. But many of these things are unanticipated modifications, and there's now like blind open PTM modification searches and so on, and people have put quite some work into these. And when they then look at what they actually find, right, using these blind modification searches, a large fraction of them are artifactual modifications that might arise from the sample prep and so on. And so the amount of, you probably discover new biology in terms of PTMs, or in terms of, some of these also look for SNPs, for example, or coding polymorphisms, or whatever. And people have had a hard time in the DDA world to buy a lot of extra value from this approach. But that doesn't mean, we shouldn't do it, right? The nice thing about the SWATH data or DIA is that all the data is there, right? So you can try and get what you can out of it. But I think it's a good point. The amount of signal that we can explain is still quite low, or not complete.
Do we still have time for questions?
I think, do we? Okay.
I have some very quick questions. Like, is there any swap databases for Orbitrap instruments or originating from Orbitrap instruments?
I don't think so.
Okay.
I mean, so what there is though is a lot of publicly available Orbitrap HCD data that you could turn into an assay, right? You could do it yourself.
Of course, you have time for it. I would like to ask the example you showed comparison between transfer from one instrument to, or from this Gillette example with 150 proteins. That was exactly the same instrument, or was it slightly different?
It was the same, exactly the same model instrument. Yeah, okay. They were both 5600. Yeah.
I see. And maybe one longer question. When you use multiple engines, do you go for the overlap, or do you take total?
So what we do actually is first we analyze each search engine with the tool, this peptide profit tool from the TPP, and what this does is puts all the scores of these onto a sort of a common framework, which is this peptide probability score, and this allows you to which is a real probability, and this allows you to combine them in a statistically meaningful way. So there's a whole paper about this iProfit interprofit tool and the method it uses to kind of sensibly combine this data, but just naively combining them together may not be the best way the most efficient way to do this. But there are tools like iProfit and there's also this commercial tool Scaffold which does a similar thing, and there are tools out there which do this. So I wouldn't just like combine the lists. This is probably not the right way to do it. Was there one more, or should we go? Yeah, I don't know. Quick one.
A quick one. Is there a lower number of samples I should collect for a library? So what you say, if I want to make my own library, I have to collect for 10 samples, or is there a limit for the beginning?
So this is still somewhat an open question, I would say. So for example, you can either take like a representative sample and inject it many times, and Lucas from Biognosis has done some work on this, who will talk on Friday, to say somewhere between kind of 6 and 20 injections you get kind of saturation, right? So if you inject the sample over and over again, because of the stochastic sampling of DDA, you don't get it on the first one, but if you inject it enough times you eventually get to saturation. Whether that's the best library to then, so that's a good start. Whether that's the best library in the end to the most comprehensive library to do the SWAT analysis, you may want to extend that library a bit deeper by, for example, doing some fractionation, which you do in the library generation step, but not in the SWAT analysis, right? In the SWAT analysis, you just do a single injection step, and because we know that the SWAT analysis should be a little bit more sensitive than the DDA. So if you just do DDA side by side on the same samples, maybe you don't get to quite a sufficiently comprehensive library to get the most out of the SWAT data. Did I explain myself? So maybe you want to make the library a little bit more comprehensive. But then there are other statistical considerations. If you make the library very large, then the statistics get a bit more difficult, and I think Yancheng will also mention something about this. But already doing replicate injections of your samples in DDA mode, up to sort of 10 times or something, I think is a very reasonable place to start, and then you can think later about pushing it down or. Okay, I'm sorry if I went over time.